Yeah. So I am recording today's meeting. It is supposed to be a live session, but I did see that the link has not been posted on the course portal. So it might be possible that people are not aware of these sessions, right? So anyway, I will record this particular session and I'll post it on the YouTube channel that I'm supposed to post it on. Along with that, NPTEL will have access to the recording and they'll share it with you. So let us begin with this session. Let me just share my screen for the same. Okay. Yeah. So I have basically divided this course into two parts. Let me begin with the first part. I'll actually switch briefly over to the second one. So, as I said, this is at least it's supposed to be a live discussion session, and this is pertaining to the course Neurobiology that you people are enrolled in. And what we'll do in this particular session is that from the previous year's assignments, we are going to solve those assignments so that it will give you a heads up, and it will help you probably in solving this year's assignment as well because the deadline for your assignment submission is not over for that particular week for which I'll be taking this course. That is why I will not be discussing the current year's assignment, but I went back and checked. There are some overlaps, or if not the question, if not the overlaps between the questions themselves, there are some conceptual overlaps, right? So what we can do is we can go through the questions that had been posted last year. It will help you increase the pool of questions that you are solving. That way, you are submitting an assignment. There are a number of questions in that. Along with that, there are previous year's questions also that you are getting to solve, which will help you understand this course better and it will also help you prepare better if you have enrolled for the exam, right? What we'll do first is I have the previous year's questions. That is what we are going to discuss first. It is in a Word file. Then we'll come back to this particular presentation and we'll discuss the overview of this week in with respect to what has been taught to you this week, right? So there were 9 lectures. I think I have been able to consolidate around 7 in this overview. The following two I'll include in next week's discussion session. Okay. So let me, as I said, just let me go back to the Word document with the previous year's assignment, right? So, yeah, yeah. So I hope my cursor is visible, right? Yeah. So the first question, this is from assignment zero. I went back and checked. It is the same assignment zero for week zero that has been given this week or this time also. And since you are not scored for this or you are not getting any points to solve this, but like you should be knowing the answers to these questions also. That is why we'll begin our discussion with them. So the first question is, which of the following structures is not present in the brain? Right? The options given for this particular question is cerebellum, pineal gland, hippocampus, and adrenal gland. Right? So which one of these are not present in the brain? The answer is simple. It is adrenal gland. Right? So people who have a background in biology will know that adrenal gland is something that is present on the kidneys, on top of the kidney, right? While even if you do not have a background in biology, the other three terms might have been things that you might have heard during the first week lectures of this particular course, right? So cerebellum, it is basically with a part that is controlling the motor functions of our body. Then pineal gland is the gland that was talked upon by Vesalius, if I'm not wrong, by Vesalius, who had this misconception that the pineal gland does not have is a single gland that is present at the junction of the two regions of the brain. Although later it was found that pineal gland is also bisymmetrical, and it is also present in two parts in both hemispheres of the brain. Then hippocampus is the part that is again, it has been discussed upon. It is named on the Latin name of a seahorse because that is what it looks like when it is extracted out of the brain. Also, this is the region that houses the place cells, or it has the ability basically to uh, locate, to help our brain in us accessing locations and in remembering locations, right? So that is hippocampus. Now, moving on to question number 2, which is, which of the following statements is the most accurate? Now, let us go through these statements. The first one is, brain does not control the heart rate, right? The second one is, brain does not control the breathing rate. Then the brain does not control the knee jerk reflex. And the fourth is, the brain does not control the vocal cords during speech. Right? So we have to find one statement that is the most accurate. Out of this, let me first eliminate the things that are not true, right? So it says brain does not control the heart rate, which is wrong, right? There are, although heart has an autonomic nervous system of its own, but it does have involvement of the brain, right? There are some branches that are going from the brain to the heart that is also controlling the heart, right? Then it does not control the breathing rate, which is false again, because uh, breathing is an involuntary function that is basically being taken care by the brain stem, right? Then brain does not control the vocal cord during speech. This again is incorrect. There are vocal centers that are present inside the brain, and we have also seen in a few uh, depictions of the brain that there are vocal sensory regions that are present. So this again is not correct. But brain does not control the knee jerk reflex. So this option is actually the correct option. Why does it not control the knee jerk reflex? Because knee jerk reflex is basically a reflex action, and in that the jerk that is caused on stimulating the patella region of the knee is actually a signal that is sent to, and it is responded to by the spinal cord, right? So there is this basal ganglionic region in the spinal cord which is taking care of that reflex action, right? The brain is not directly involved in that. So this is the correct option. Then the third question is, which of the following scientists received a Nobel Prize for research related to the brain? Right? So your options are Venki Ramakrishnan, Eric Kandel, Svante Pabo, and Shinya Yamanaka. Right? So all of these are great scientists. I'm not sure if we have studied this yet, that which one of them have actually received the Nobel Prize because of their work related to the brain. But even if we have not studied it in week 1, it comes, I think, in week 2 lectures. And the answer for this one is Eric Kandel, right? So what did Eric Kandel do? He basically studied how signals are transduced or signal transmission across neurons is taking place, right? So the correct answer for this one is Eric Kandel. Then question number 4 is, a mosquito has a brain. Is it true or is it false? Right? So this is true. Even the primitive organisms, they do have a brain, right? Mosquito also has a brain. It has a network of neurons, and there are different the kind of neurons might be a little different, and the organization of the brain might not be same as that in mammals, but it does have a brain, right? So moving on to week 1 assignment that was given in the last year, and it is kind of similar. Some concepts are kind of similar to what you have been assigned this year, right? So we are going to see that. Just a moment, just let me have water. Okay. So the first question is, which one of the following organisms has the largest number of neurons, right? So is it the chimpanzee or the human or the elephant or the C. elegans, right? So the answer for this one, it is pretty simple. It is the elephant, right? So we had this discussion when Sir has taken this course. He has had this discussion while he was discussing that what is the basis of intelligence, right? Is it the number of neurons? So we did conclude that it is not the basis of intelligence is not due to the number of neurons because in fact the although the most intelligent individual or organism out of all of these is the human, but the largest number of neurons is present in the elephants, right? So elephants have about 250 billion neurons, while humans have only around 86 billion neurons, right? So the largest number of neurons are present in the elephant out of the ones that were given in the option. Then question number two is, fMRI has a better temporal resolution than EEG. Is it true or is it false? So fMRI is a functional magnetic resonance imaging, EEG is the electroencephalography, and the question is which one out of these has a better temporal resolution, right? Is it the fMRI? So the answer is basically false because fMRI, sorry, because EEG includes electrical measurements and it has better temporal resolution than the fMRI, right? So fMRI, the recording is how you can understand or remember it is that in EEG, where are the electrodes? It is placed directly on the skull, right? So it is capturing the data that is coming right from the skull, from the brain, crossing the skull, and it is being recorded, right? While in fMRI, you are going into an MRI scanner, right? So the distance between the detection and the source is also more, right? So even like with that basic logic, you can answer this question. Moving on to question number three, which says, which of the following is not a part of a typical neuron, right? Is it a nucleus or an axon terminal or a microglia or myelin sheath? So to answer this, let us go through what these components are. What is a nucleus? It is basically the cellular organelle that basically contains the DNA or the genetic components that is present in any cell, right? So a typical neuron, like most of the cells, also has a nucleus. Then an axon terminal. Axon is actually one of the characteristic features of a neuron, right? So it is uh, it is an elongated sheath or an elongated basically uh, fiber that is coming out from the cell body of the neuron and that is helping in transduction of signal from one region of the brain to the other region of the brain or the body, right? So axon is present in the neuron, thus it also contains an axon terminal. Then uh, comes the myelin sheath. Now myelin sheath is also a typical of a neuron, but not all neurons might have it, right? It is a kind of an insulation that is present on the axon terminals, right? It is formed by oligodendrocytes, which are the glial type of glial cells that are present in the brain, right? So it is also present in a typical neuron, but not all neurons have it. While microglia is a glial cell that is different from a neuron, right? A neuron is a neural cell, then there are glial cells, right? One of the glial cells is the microglia. It is actually the resident macrophage that is present in the brain. So microglia is a different cell type altogether. Thus, the answer for this question is going to be microglia, right? So microglia are a type of glial cells and they are not a part of neuron. Okay. Question number four. If a person suddenly comes across a lion in a forest, the autonomic nervous system that is likely to get activated is the sympathetic nervous system. Is this true or is this false? Now, this is something that is not, I think it is not new to most of you through this course, right? We know that our sympathetic nervous system is something that is responsible for our fight or flight response, right? So it will be true over here because sympathetic nervous system is going to stimulate the fight or flight response, and over here it will be a better decision for a person if they choose flight over fight, right? So uh, that is going to be true. This question is going, the sympathetic nervous system is actually going to be activated. And if, for example, the person chooses to run away or the flight response, right? After it has gone or reached a village where there are a lot of other humans that he has encountered, and now the person has a safe place to stay at, right? So at that time, the sympathetic nervous system, whatever stimulations it had performed, that will go down, and he will come to more of a calm and a rest state. And in that, the parasympathetic nervous system will be involved, right? Then question number 5 is, which of the following best describes the location of the primary visual sensitive area in the brain, right? So this is a tricky question because you do need to remember that rough map of the brain that had been shown to you in one of the lectures, right? And then also you need to remember that what is meant by anterior, what is meant by dorsal, what is meant by posterior, ventral, etc. Right? So first let us come on the terminology. So anterior or rostral means the front side that is facing like the eyes or the face, right? That is the anterior or the front side or the rostral side, and posterior is the side opposite to that. So that is anterior and posterior. Then what is dorsal and ventral? So a simple way to remember this is uh, your back side is your dorsal and your front side is your ventral, right? Or when we did not used to be bipedal, right? For example, if we take a dog, the dog is not a bipedal animal. It walks on four limbs, right? So whatever organs are facing or whatever regions are facing the sun are basically the dorsal regions, and whatever is facing the ground is a ventral region. So over here, dorsal and ventral means the head portion is the dorsal and this portion is the ventral, right? This part of the brain that is facing the ground. Then lateral means on the side. Right. So now that the terminologies have been cleared, you still need to recall that brain map that was shown to you. And the answer for this, the primary visual sensitive area in the brain is in the posterior ventral side. Right. So it is the present in the occipital cortex, which basically contains this primary visual processing area in the brain, and that is located on the posterior ventral side. Right. So what will be the posterior ventral side means at the back towards the bottom, right? That is where the occipital area is, and that is where the visual processing and visual sensitive area is, right? Coming to question number 6. So question number 6 is, which of the following brain areas is most involved in keeping track of your location in the world, right? So your options are amygdala, hypothalamus, medulla oblongata, and hippocampus. So, which of these is the most involved in keeping track or location wise, which is the most involved area of the brain? So, as per studies up till now, what we know is hippocampus is most involved in the location related tasks, right? While medulla oblongata is involved in most of the involuntary functions, right? Taking care of them. Hypothalamus is something that is involved in um, sense of temperature changes within the body and other, regulatory mechanisms or feedbacks that go on within the body. Amygdala is the center that is majorly involved in stimulating of fear response, right? Emotion, it it stimulates the emotion of fear, right? So that is what amygdala is doing. So the answer for this one is hippocampus. So it is involved in the spatial navigation and it has place cells. Now these place cells are the ones that have been one of the Nobel Prize rewarding discoveries in the past decade, right? So we come to question number 7, which says in his famous experiments, Penfield measured the electrical current generated in the brain when the patient moves specific muscles. Is it true or is it false? Right. So you have to recall who was Penfield. Right. He was the doctor, the Canadian doctor who was basically mapping out brain regions, right? But did he measure how much electrical current is generated in the brain? No. So this is false. On the contrary, what he was doing was he was once during craniectomy or during the brain lesions removal in epilepsy patients, where once he opened up the brain, what he was doing was in these subjects, he was stimulating different brain regions and he was checking what kind of response that person is getting, right? So he did not measure the currents. Instead, he stimulated different parts of the brain and observed effects of this on the body, right? Coming to question number 8, it says bipolar neurons have two main dendrites and one axon, right? Is it true or is it false? So it is actually false because bipolar neurons have one dendrite and one axon that is coming out from the cell body, right? So they have one dendrite and one axon. In the presentation that I will show you later on, or I'll discuss with you, I will explain like how to remember nicely what is the difference between unipolar, bipolar, multipolar, etc. Right? So, coming to question number 9, Golgi found that the brain is composed of different neurons. Is it true or is it false? So, what did Golgi do? Camillo Golgi, after whom the Golgi complex is also named, right? What did he do with respect to the brain? He actually developed a staining protocol for the brain, right? He developed a silver staining technique, which is still date it is used for the histological study of the brain, right? Because it is taken up by some neurons and it helps in differentiating that in the mass of the brain, right? So that is what he did. But did he find that it is composed of different neurons? On the contrary, what Golgi thought was that after he stained the brain and he drew the cerebellum or the hippocampus, what he said was that the brain, unlike other body organs, is not composed of different components. Instead, it is like made of a continuous network of a single cell type, something like that, right? So, this in fact is false. He believed that the brain is a giant interconnected tissue and did not observe different neurons. Who observed the different neurons using his staining procedure? It was Kahal, right? And later, both of them were actually awarded the Nobel Prize for their Golgi for his staining technique that he developed, and Kahal, who actually made the logical conclusions based on what he saw or what he observed using those staining techniques, right? Then coming to question number 10, which of the following techniques has the best spatial resolution? So which of these techniques has the best spatial resolution? Is it the functional magnetic resonance imaging, patch clamp electrophysiology, electroencephalography, or positron emission tomography? Right. So out of all these, patch clamp electrophysiology actually has the best spatial resolution. What is meant by spatial resolution is that uh, how minute in detail can it actually detect something, right? So it does not ask that how effectively it can detect a frequency. No, how small region can it actually detect? So patch clamp electrophysiology is usually used on single cells and sometimes even on single receptors on those cells, right? So that is why the answer for this one is patch clamp electrophysiology. The others have a higher spatial resolution, which ranges from millimeters to centimeters. Patch clamp, on the other hand, it will be in microns, right? Because a micropipette is being used to go and attach on basically the surface of a single cell, and reading is being taken from that particular cell using this technique, right? So, the patch clamp electrophysiology can measure and the activity of the individual neurons or even specific parts of a neuron at the resolution of about 1 micron, right? While other techniques cannot. Their resolution is generally from millimeters to centimeters, right? So, yeah. So that brings us to the end of the discussion for the previous year's assignment. Uh, I'm going to minimize this and we'll come back to our presentation, right? So just let me take a okay, moment. I have stopped presenting. I'll start presenting again so that there's no confusion, because I don't really have people over here who can tell me if it is not getting recorded. Um, yeah. So I hope at least the practice session would be useful for you guys once you actually see that. Now we'll come to the live discussion session, right? So in this, what I have done for this week is that I have kept a recap for what has been taught to you in week one by Sir in his course. Along with that, there are some points wherein we have elaborated more, like for instance, the techniques for studying the brain, right? So I have elaborated a little more on each technique, still not going into much detail about all of them, as not to keep this lecture towards a very, intellectually draining side, but a slight more emphasis on each of those techniques, because I also want you guys to learn a little more. If you are like attending an extra session, there should be some extra knowledge that should be imparted, right? So having said that, let's just begin with the session. Also, my apologies, because the learners had not enrolled. I actually forgot to introduce myself properly. I'll take a moment to do that. So I am Arveen Kaur. I am a PhD scholar at Department of Biological Sciences and Bioengineering at IIT Kanpur. Sorry. Along with this, I am also a PMRF scholar and a TA for your course, that is neurobiology. The This particular presentation will include material from Professor Nitin Gupta's NPTEL lectures, and also some other internet resources and research articles which I have mentioned as I have gone along the lecture. Right. So let us begin with it. This is just a brief introduction for you guys. That what we are basically going to talk about in this session is linked in some form to either the human brain or understanding of the human brain, right? So human brain roughly weighs about 1.5 kgs, and it is comprised of three main regions, that is known as the cerebrum, which is the largest region of the brain. Then comes the cerebellum, and then comes the brain stem and the medulla oblongata, right? Starting from the very basics, I have also summarized the historical timeline. Sorry, just a moment, guys. Yeah. So I have also summarized the historical timelines that have been taught to you in this particular lecture, which begins in 500 BC, wherein Alcmaeon of Croton, who is a Greek philosopher, basically he worked on he worked by cutting the nerves of certain animals and seeing what is happening. And based on that, he determined the brain is the seat of the mind, right? So the brain is the central controlling region that is present in our body, right? Then in 400 BC, after him, Socrates, who is also known as the modern father of modern medicine, agreed that the brain was the seat of intelligence. Thereafter came Aristotle, who is also a very famous philosopher, but he, on the contrary to Alcmaeon and Hippocrates, what he thought was that heart was the seat of intelligence, and what was the brain doing? Brain was actually present to cool down the mechanism that the heart was running, right? So he thought that the humans were superior than other animals because it had a cooling mechanism for the heart that was actually running the entire body, right? Then even in India, people in 200 BC, there are records of people who were thinking about the brain, and one such record is found in Charak Samhita, and the author of this was Charak, and he basically said that if the brain is injured, there could be many body functions that can be affected or compromised, right? So there could be blindness, there could be impaired motor functions, impaired speech, etc. Thereafter, in 150 AD, Galen, he basically said that the spinal muscles control specific muscles, right? Spinal cord, sorry, controls specific muscles. The cerebellum controls muscles, and the cerebrum processes the senses, right? All these three are kind of correct. So even in 150 AD, when most of the techniques that we use now to study the brain were not, available or even thinkable at that time, Gillian was able to make these conclusions based on observations that he had from his patients, right? Then brain functions by movement of animal spirits through ventricles. This was an additional statement that he had made. So, what are ventricles? Ventricles are basically the hollow regions of the brain that is seen in the MRI, or once you cut open an autopsy brain, there are some hollow regions that are visible. That is basically the ventricles. But mm, like in an intact brain, they are not actually, hollow. They are filled with uh, fluid that is known as cerebrospinal fluid. So he thought that instead of the cerebrospinal fluids, it was actually the animal spirits that was flowing through these ventricles, which was also maintaining the function of the brain. Right? Thereafter, we skip far ahead and we come to the 16th century, wherein Andreas Vesalius basically identified many structural features of the brain and concluded that brain functions through various nerves. Right? So this was also a fair assumption, and it was actually correct. Later in 16th century, Leonardo da Vinci also built upon Andreas Vesali's works, and he made detailed sketches of the brain, right? Leonardo da Vinci, again, is also a very famous artist, and he was also interested in science at that point, right? So he made detailed sketches of the brain, which again give insights to the philosophers that came and after him, right? Then in 16th century, René Descartes, who proposed basically the theory of dualism of mind and body, that mind and body were separate and they were connected to each other at the pineal gland. So that is what Descartes said, right? Then he also said that this pineal gland controls movement of muscles through CSF in the ventricles, right? So again, cerebrospinal fluid is coming into picture, and it was believed even by Descartes that this cerebrospinal fluid is actually what is controlling the movement of muscles, right? Later in 1780, Luigi Galvani, who was working with electricity, he basically showed the role of electricity in nerve conduction and muscle activation. Right, so he was using frog skin basically to generate some kind of electricity, and accidentally a dissected, opened, and dead frog that was lying on the table happened to come in contact with the source of electricity. And what he saw was that as soon as there was electrical contact with that dead frog, its muscle twitched, right? Even though it was dead, it did show some contraction and expansion of the muscle, right? So that gave insights to the scientists that came after him that yes, there could be role of electricity in the muscle in the nerve conduction, right? In 1850, Helmholtz basically said that neural activity is electrical in nature. So he definitely concluded this, and he designed experiments to measure the velocity of the signal propagated along the sciatic nerve of the frog, right? So we are going to actually discuss these experiments later in detail further in this course. Thereafter came Camillo in 1873. We just read about him in our practice in our assignment that he basically developed silver salt based staining technique for the neurons. Although he claimed that the entire brain is a single large network, right? But the silver salt-based staining technique, nonetheless, was a gold standard technique. It is still being used till date for histological studies. And after him came Santiago Ramon y Cajal, who actually took the most advantage of this technique, and he was able to visualize various different cells in the brain, which were different types of neurons, right? So that was a brief timeline that you have studied through the first and second uh, units of this particular week, right? Then came the neuron doctrine. There were actually three doctrines. We are going to go through all three of them. As I said, Ramon Cajal, who is also known as a father of modern neurobiology, right? He basically just through staining and observation and recording of that observation through drawing, because at that time we did not really have cameras attached to the microscope. So he was a great observer, and he was also a great artist. He could draw whatever he could see, right? Based on that, he concluded a few things, right? One of them is the neuron doctrine, which basically says that the nervous system is made up of discrete individual cells, which are known as the neurons, which are supported by astroglia and by other glial cells, right? So at that time, he could make out different types of cells in the brain. Out of that, he could make out different kinds of neurons and other cells apart from the neurons as well, right? So that was a great achievement at his time. So next we come to the other two doctrines that were proposed by Ramon Cajal. The first is the principle of connectional specificity, which says that the nerve cells do not connect to each other randomly to make a network, but the connections are very specific, right? And this is actually true. During our development, there is a whole process in which the correct connections are being made, and after it happens definitively is when we start evolving, and our memory starts developing, right? So principle of dynamic polarization was the second doctrine, which says that electrical signals within the nerve flow in only one direction, from the receiving sites of the neuron to the trigger region at the axon, which is also true. The flow of signal is not bidirectional, it is unidirectional, right? And again, the emphasis to be laid on is on the fact that he could make these observations even though he was not doing any functional studies. He was just staining the tissue and observing what is the outcome of that staining, right? So, coming to how complex the human brain is, right? So now that we have briefly revised what the history of neurobiology and neuroscience was, let us come to uh, account for the complexity of the human brain, right? So it does have 86 billion neurons and almost about a similar number of glial cells. So those many number of billions and billions of cells are actually present in the brain. The number of synapses are no less, right? There are around 10 raised to power 14 synapses that are present in the brain. Along with this, thousands of neuronal cell types and other cell types are present inside the brain. The capacity of human brain is around 1400 G or 1400 cc, as was shown in one of the comparative slides or the evolutionary slides with the chimpanzees and the apes, right? And it has more surface area due to the presence of the sky and the gyri, right? So more ridges and folds are there in our brain, which basically increases the surface area of our brain, right? So the effectively the amount of things that can be packed into that much area is increased because there are a lot of sulci and gyri, right? Then we come to some primitive techniques of studying the brain, one of which was phrenology. Phrenology is basically the study of the conformation of the skull as indicative of mental faculties and traits of character, especially according to the hypothesis of Franz Joseph Gall, who was a German doctor. And in 19th century, there were other people who were supporting his hypothesis, right? So what is the basically phrenology is that people or scientists in early 18th century, what they believed was that if you study the skull of a person, right? So, for example, if somebody had some kind of a ridge on the skull, or it was bulged at some point, or it was hollow at some point, right? It could actually tell what is the kind of, what uh, perspectives that that person has, the kind of instincts that that person has, whether the person is a genius or he's a con man, etc. Right? So people actually used to believe that the study of the skull can actually reveal these traits about a human being, right? But in fact, it is not true, right? Which was understood later in the 18th century and in the early 19th century, and that is when this phrenology was stopped being used, right? So it is not in general practice right now. But since then, since we actually had to still study the brain, there were other things that were coming up, right? How to study the brain. Like, we don't know what the brain is, how it is functioning, right? Okay, we now know that it has a number of cells and our brain is bigger than the monkey's brain, etc. But we still don't know how to actually determine which region is accountable for which function, right? So that came much later with the development of these techniques. Right, that is shown over here. So I am not going to explain this table again because that is something that has been explained to you by Professor Gupta in his slides. But I'll just pick up these individual techniques and I'm going to discuss about the techniques, right? So the first kind of techniques that came to emerge were the crude techniques that were the lesions and micro lesions. Now, why were these techniques crude was because in this what was happening was that there were naturally occurring lesions or regions in the brain that were not actually functioning or that were creating some problem in the functioning of the rest of the brain. Right? Such lesions or smaller size of lesions, which are known as micro lesions, were actually naturally present in the brain, right? And they could not be cured. So, what used to be the treatment for that was that the person's brain, the person who was suffering from these lesions, so their brain used to be opened up, and that particular region which had that lesion or which was the problematic part in the brain, it was removed basically through surgery, right? And sometimes it was a little region, it would not cause much of a change in the person's behavior or the phenotype or how the person is basically responding to things or whether he has dementia after that or not, etc. Right? But sometimes the lesions were either too big or they were located in some specific regions which were associated with some specific function, right? So what was happening was that if, for example, I have a lesion in my hippocampus, so if that area is removed, I will probably not have any sense of location, right? Maybe somebody told me to come from like the institute to my home and I will not be able to do that, right? Or maybe somebody just directed me that, okay, you are seeing that road, just go straight and take a left, right? Or take a right. I might go straight and my brain might not know what a right is or at which point I had to take a right. Okay, so my comprehension might come down to that level, right? That I might not remember any spatial cues. So, if my hippocampus had a lesion, which was basically removed, right? But since that was the only treatment at that time, a lot of these lesions were actually removed, and the doctors that were, or the neurosurgeons who were doing these surgeries, actually kept records of these, right? So they were also curious people and scientists. And whenever they were removing some particular region, they were keeping a record of where that, what part of the region of the brain it was representing, and what was the outcome that the patient was facing after that region of the brain was gone, right? So some examples are given over here, which is from a book that I have referred over here, which basically talks about language mapping, right? So over here, it is actually fMRI that has been used to look at this. But there was also intraoperative stimulation, which happens when people open up for lesion, right? So it was seen through both these techniques actually that once for example, in intraoperative stimulus, if the this Broca's area region was stimulated in the brain, or if it was removed as a response, as a treatment for it being a lesion, right? Then people's speech used to arrest during counting, right? Yeah, randomly in the middle, it will arrest and we'll not be able to initiate that again, right? Then there was this area, which is referred to as the Wernicke's area, which if was removed, there was literal paraphasic speech error during picture and naming, right? Also, there was this other area which, if it was stimulated or if it was removed, the person would have difficulty in finding words during picture naming, right? So you might show them a placard and they might know that, okay, this is a picture of a dog, but they might not be able to find the word for the dog word inside the brain, right? So that will be impaired, right? So that is how, initially these throat studies were performed, but they have actually helped our knowledge about what region of the brain is doing what function up to a great degree. Okay. Yeah. So there is this very recent paper from Nature Communications into 2019 that I have also kept over here. It is titled Human Memory Circuit Derived from Brain Lesions Causing Amnesia. So in this, they have basically taken the records for thousands of brain, and I think they have screened, finally after screening, they have assessed 53 brains, and according to what regions the brain lesions were in, they have they have basically mapped it out with what is the general sensory circuit that is followed by a person. And they saw that, yeah, in around 51 out of 53 of these regions that were screened out, it was seen that the circuit of the brain, that the somatosensory circuit that the brain is involved in some specific function, it always passes through a particular point in the hippocampus. And 51 out of these 53 brains actually had a lesion in some part of the circuit, so that it could not reach that part in the hippocampus, right? So they had problems in basically forming of memories, which also the hippocampus is involved in. So, like, it is not for you to make you understand what the study is. It is more like an example to make you understand that these like data from these kinds of studies and records are even still being used to understand key functions of the brain, right? The second one is the PET, or positron electro and tomography, right? And along with that, 2-deoxyglucose, right? So 2-deoxyglucose, along with other kinds of contrast agents and fluorophores, is also one of the or autoradiographic fluid that is used in the PET, right? So we are going to talk about both of them together. So I'll just have a water break. Yeah. Okay. So this 2-deoxyglucose, or 2-DG technique, was originally developed for laboratory animals by Louis Sokolov, right? And it was later adapted to PET for humans, and has been provided, and it has provided a direct demonstration for the coupling between neuronal activation and glucose, right? So, prior to coming to what this technique is doing, let me give you a brief introduction about glucose and brain, right? So, glucose is a primary energy source in the body, right? It is a carbohydrate, it is the primary energy source in the body, and it is actually the only utilizable form of energy source that can provide energy to the brain, right? So, brain, although it is uh, like one of the body organs, it is almost consuming about 20% of the ATP that is generated by glucose in our body, which is a huge percentage, right? So that itself tells how important this particular organ is, right? So over here, what is happening is that people were trying to understand the coupling between the glucose consumption and the neuronal activation, right? So this 2-deoxyglucose was basically a homologue of the glucose, and this 2DG technique is an experimental autoradiographic method that measures the regional brain glucose utilization, right? So once it is sent into the brain, what will it measure? That uh, uh, how it will show up is that the regions that are utilizing more glucose will give a particular fluorescent or more, heated colors in that autoradiographic analysis that we are seeing. And the ones that are not using that much glucose will give cooler colors like blue, green, while the ones that have a lot of glucose utilization might be red, or reddish yellow, etc. Right? So it can also be used to study neuronal activity and glucose coupling, right? So, okay. So this is an illustration of a PET scan, right? So it is the like the scanner, similar to the CT scan and the MRI machine. which is attached to a monitor. It has a rotating scanner and a motorized exam table, right? And this is the kind of output that it gives. So as I said, in this orange reddish regions are the ones that are actually using the most glucose, and these blue regions are the ones that are using the least glucose, right? And let me just show you. Yeah. So uh, this SUVs basically mean the standardized utilization value of the glucose, right? And you can see that the PET analysis of the brain somewhat looks like this, right? So in this also, what you will see is that if there is a normal brain, right, there will be a consistent metabolic activity throughout the cerebral cortex, right? Cortex is these outer regions of the brain, right? Then, in Alzheimer's disease or in some other disease like frontal lobe dementia or Pick's disease, whenever there is a pathological condition of the brain, there will be reduced metabolic activity, whether it is in temporal and parietal lobes in case of Alzheimer's disease or in the frontal lobe in the Pick's disease, right? So we can clearly see a very stark difference between the normal and the pathological conditions, right? That is also one way how to assess whether there is some pathology that is going on in the brain, or is it a normal healthy brain, right? So it is one of the measures for that also. Coming to the other technique, which is fMRI or functional MRI, it is a type of MRI scan that shows which areas of the brain are most active. Again, it is similar to that 2DG technique that we were doing earlier. Right over here, what is being practiced? They are tracking and comparing that activity to what you were doing at the time. At the time, can help map your brain activity. It's most often used for planning surgery or similar procedures in the brain. Right over here, what is actually being monitored is the oxygen that is being used. Okay, so first let us see what a standard MRI does. A standard MRI scan uses extremely powerful magnet, like thus the name magnetic resonance imaging, radio waves and computer processing to generate highly detailed 3D pictures of the inside of your brain. Okay, this is done by an MRI scan. What is an fMRI doing? It is using the same MRI machine, but it is special because it is tracking the blood flow in different parts of your brain, right? And this blood flow is basically correlated with how much of oxygen is being used by these brain parts, right? So if it is more active, it will require more blood flow because it will be using more energy and more oxygen, right? So that is the principle of fMRI. So, in a structural MRI scan, this is kind of the output that you get, and in a functional MRI, you can see that there are some regions that are lit up, right? That is the kind of output you get. So, structural MRI is a non-invasive technique for examining the anatomy and pathology of the brain. Right. So if for example, this was a demented brain, even in MRI, I will be able to see areas that, okay, this area has less of gray matter or white matter, or there is tissue that is dying over here, that I'll be able to make out from a structural MRI also, right? Or if there is a bulge or a lesion, I will be able to make it out over there. But in functional MRI, it is non-invasive technique for examining the brain activity, right? For example, if I am getting, or if somebody is getting nightmares, right? It can be monitored through fMRI. How, like, often, or what is the region that is most often being lit up in that person while he or she is getting a nightmare, or in epileptic people, like what is the area that is having abnormal brain activity, something like that. Okay. So so during different tasks being performed, different areas will be lit up in your brain. So in an fMRI, if for example a person is speaking, okay, these are the areas that are utilizing most amount of oxygen, right? The blood flow is maximum in these areas that are lit up in yellow. When a person is tapping their fingers, right? For example, if I am using a mouse and I'm tapping my finger on it, maybe this part of my brain will be lit up, right? If I'm listening, then these regions of my part might be of my brain might be lit up, right? So um, the point is that fMRI and PET scan are kind of similar, right? Both of them are giving us an output regarding the brain activity, right? So the question arises as to which one is a better technique, right? So I would urge you guys to go and read about it. But there are a few points I would like to discuss right now also. So, in positron emission tomography, what we are doing is that we are basically injecting something, and then that is basically producing some radio labeled or radiograph, something that is being able to be detected by the tomography machine or the X-ray detector that we have, right? In fMRI, however, it is the innate activity of the brain and nothing needs to be injected over here. So it is generally said that if you have like one scan that has to be performed, uh, it might be more probable that the spatial resolution is better in case of the PET scan, right? Because in that you are actually giving a homologue and it is being utilized and it is a real time thing that you can see. But if you can afford it, and if it is, for example, for an experimental purpose, it is favored to do fMRI because over here there is no harmful substance, like even though that is not toxic per se to DG, but it is radio labeled, right? So anything radio labeled, if you are injecting at higher amounts or if you are injecting it again and again, it will not be good for your body, right? So if you need to repeat experiments again and again, it is favored to do fMRI. And because you can have a larger sample set or larger data from the fMRI, compare and you can conclude based on the comparative analysis of all this data. Based on that, it is said that fMRI is better, right? So I hope you understood the gist of it. Okay. So coming to the other techniques, which are very similar again, they are MEG or encephalography, ERP or event-related potential, and EEG or electroencephalography. Okay, so let us first talk about MEG or the magnetoencephalography. It is basically a non-invasive medical test that measures the magnetic fields that are produced by your brain's electrical currents, right? So we know from whatever physics that we have studied that whenever there is a current that is passing through a particular wire or a particular place, right? That current will stimulate the region around it and it will have some magnetic field around it, right? So this magnetic field is what is being detected by this MEG procedure. It is performed to map brain functions and to identify the exact location of source of epileptic seizures, right? Then comes event-related potential, which is ERP. The methodology captures electrical activity instead of the magnetic field. It is now capturing the electrical activity in the brain at specific time points related to an event. So ERP is event-related potential. In this, the major uh, point to remember is there is always a stimulus that is given. It could be a visual stimulus. Somebody might be showing you some particular kind of an image and might be changing those images continuously. Or some other kind of stimulation, but there is always a stimulus that is preceding this recording, right? Then comes the electroencephalogram or EEG. It is basically a test that measures electrical activity in the brain. It is also called an EEG and it uses small metal discs, which are known as electrodes, that attach to the scalp and then the brain cells communicate via impulses and this activity shows up as wavy lines on EEG recording. The brain cells are active all the time, even during the sleep, right? So in this also, as we were seeing an ERP, again, it is an electrical recording only, even in this case. And in this, the recording is done via electrodes. And the major difference between EEG and ERP is that this is a spontaneous recording of a recording of the spontaneous activity of the brain, while in ERP, there was a stimulus and the response to that stimulus was being recorded from the brain. It was not a spontaneous activity of the brain, right? There are depictions of all three that I have kept. MEG, like I said, if there is an electric current that is passing through, there will be a magnetic field around it, which is being detected in MEG, right? So the intracellular current is actually coming from the dendrites, and a cluster of dendrites at one region will basically give out the magnetic field. So this is the source. Then the orientation of the neurons will actually determine the magnetic field, and there will be a detection device which will be placed a little at a little distance from our skull, right? And then the magnetic field patterns will be recorded, and these models have to be understood, and the results will be computed, right? Then comes ERP. So as I said, in this, the key feature to remember is the stimulus, right? So, and usually the stimulus is a visual stimuli, right? So there will be a stimulus, and the electrical activity from top of the skull will be recorded. There will be an amplifier and a signal averager, and it will give us an output in the form of such a waveform, right? Then comes EEG. Again, as I said, it is very similar to ERP. The only difference being that it is calculating a spontaneous brain activity, right? So there will again be electrodes that will be placed on your skull, and a similar setup as ERP, and the brain wave patterns will be obtained, which can be processed and understood. Then comes optical dyes. Okay, so optical dyes is something that is generally not used that much in the humans, right? It is more often used in understanding of brain in animal models, right? So optical imaging is generally done by a multiphoton microscopy and can measure activity dependent changes in the fluorescent signals directly from individual neurons, astrocytes, and blood vessels in the living brain, right? One of the techniques that has come up in association with this is the optogenetics, in which basically the calcium signal is being detected by creating a cranial window in the mice, right? One of the major techniques under this optical dyes is a voltage sensitive dye imaging, or the VSDI, right? In this, there is an accurate imaging method that uses small intensity changes of the fluorescent dyes emission to capture the brain activity. Right. So, as I said, it is generally, or more often, it is used in animals. There might be very few cases of human application in case of optical dyes. And what is being done is that there is some kind of stimulation. It could be something that has been done to the mice, or some feed that has been given to mice, and the mouse is feeding, etc. Somehow the neurons are stimulated, and then um, uh, based on some sensitivity to the ions that are inside and outside of the cell membrane, there will be these voltage sensitive dyes that will light up, and there will be a signal that will be emitted, right? And that signal can be recorded, and it can be seen which part of the brain is giving out the signal. So, for example, if you are giving the mouse some feed, right, and that mouse is feeding on it, you know that, okay, this is the center that is responsible for signals that are going to the stomach or going to the mouth to basically feed on that particular subject. Okay, if for example you are showing something to the mouse, and it is comprehending something, and another region in its brain is lighting up or giving out the signal, you know that, okay, this is what is responsible for the visual sensory signals, right? Or processing. Then comes slide microscopy. Now, again, like optical dyes, this is also more often used in animal models than in humans. There are different kinds of light microscopy techniques like point scanning confocal, in which there's a particular laser that is basically going and exciting a particular region, and that is being the emission from that is being captured. Then there's two photon point scanning. In this, instead of a single particular laser, there is actually an infrared and multiple exciters, and it is basically exciting a single point and basically again capturing the output. Then there's standard light sheet microscopy, in which the entire plane is being illuminated at one end and the output is being detected. And then there is this swept confocal aligned planar excitation, or the scape, which is a very new technology, which is an improvement over confocal, and it basically can give a better result for larger samples, or bigger or thicker samples, right? So I'm not going into much detail of this. If you are interested, you are always most welcome to go up and study about it. And if a question arises, I can solve that. If you post it on the discussion forum or ask me, anything is fine. Sorry. So then we come to the single unit and the patch clamp techniques. Okay, so these are the final techniques that I'll be discussing. And then I'll move on to the other segment. So in unit recording, as the name suggests, it is a precise method of recording the electrical activity of the individual neurons in the brain. And it involves using fine wire electrodes to capture axonal spikes from neurons in response to various stimuli, such as visual, auditory, or somatosensory inputs, right? So, what happens in a single unit is, I have actually kept this particular image because it was very easy to make you understand from this. In EEG, what we were doing, we were placing the electrodes on top of the skull, right? So, on top of the skull and on top of the skin, right? So, this is a neuron. Then we have the layers, other layers of the brain, etc. Then we have the bone, that is the skull. Then we have the skin, and on top of that is the electrode. That is EEG, right? Then comes ECOG. ECOG is another technique that we have not discussed over here. It is a slightly different from EEG in that the skin and the skull is removed, right? So there is a small hole that is created in the cranium or the skull, and then the electrode is placed on top of the brain. It is still on top of the brain, but it is below the skull, right? So it is a little closer to the source. So that is ECOG. Then is the FPN unit spikes, right? So, which is the single unit. So, in this, the electrode is taken up close to the neurons, and then it is measured. And then is an intracellular measurement, which is what we will see in the patch clamp. That measurements are taken from within a neuron, right? So the patch clamp technique is a generally recognized approach to study ion channel function. During a patch clamp recording, a micropipette containing a recording electrode connected to an amplifier and filled with electrolyte solution is brought into contact with the membrane of an isolated cell. Okay. So again, so like we saw in the final depiction of the previous image, it is basically going and attaching on the cell. And sometimes we create suction after this attachment so that the cell membrane is basically broken at that end, and the intracellular recording can also be taken, right? But if it is on the cell and on a single channel, such a recording can be obtained. When the channel is open, it will look like this. If it is closed, it will look like this, right? This is the membrane potential basically, and this is what the recording looks like. So this has a great spatial resolution that is present. Okay. Uh, then we briefly discover what the ideal technique for visualizing brain and its function will be. So it will actually be context dependent. It will be dependent on what is the kind of feature that you want to observe. And based on that, you will pick and choose which one you should be doing, right? But technically or ideally speaking, such a technique does not exist, which is the best possible one. And if such a technique were to exist, would be some of the qualities of those techniques, that it would have millisecond temporal resolution. That is, it will be very fast in detecting whatever, millisecond or microsecond changes are there in the brain. Then it will have micron spatial resolution, like that is present in the patch clamp, and it will have unlimited bandwidth. That is, even if there is a lot of data, it can be computed, and it will be non-invasive, right? So, for example, in the spatial resolution, even though you are going up till the single neuron, but it is invasive. You will have to do a surgery, create the cranial window, etc. Right? So it will be non-invasive, so that it will be more easily doable. Right? It will be more feasible a technique. Then so I'll talk about some subfields of neuroscience and neurobiology. I have just listed that for your revision sake. They are developmental neurobiology, cellular and molecular neurobiology, system neuroscience, cognitive neuroscience, psychology, psychiatry, neurology, theoretical neuroscience, and philosophy. And I'm sure if you have gone through Sir's lecture, you will all be aware of what these particular fields are, right? Because he has discussed that in detail. Then I'll come to the cell types that are present in the brain. I'll just take a short break. Yeah. Just a moment. Any- Yeah. So there are two major types of brain cells that are present. They are the neurons and the glial cells, right? So the neurons are the non-neuronal cells, or the non-neuronal cells are basically referred to as glial cells. So we all know what neurons are, and we are going to discuss what are the different types of neurons and how they look like, etc. Just a moment, yeah, what these different types of neurons are and how they look like, right? But first, let us go and discuss about these glial cells that less of you might be aware about, right? And in this particular discussion, I have included few more types of glial cells that were not discussed in the lectures, right? So these are some of those that are depicted in this particular representation. So the types of neuroglia over here has been segregated as the central nervous system, the cells of the central nervous system, and the cells of the peripheral nervous system. We'll come to each one of them one by one, right? So the first one is the microglia, right? So this is the brain's immune cells, and they serve to protect it against injury and disease, right? So these are the resident macrophages that are present in the brain. They are known as microglia, right? All the other cells are known as macroglia, right? The first cell under macroglia is the astrocytes. Now, astrocytes are these star-shaped cells that maintain the neurons network, neurons working environment, and they do this by controlling the levels of neurotransmitters around the synapses. They control the concentrations of the important ions like potassium and provide metabolic support, right? Apart from that, they are, cells that are involved in many, numerous functions within the brain. They are also involved in inflammation or neuroinflammation inside the brain. Then these astrocytes are also lining up the blood vessels inside the brain. So they are a part of the blood-brain barrier. That is one of the key features of the brain, right? So there are multiple functions that these astrocytes are doing, right? Then comes the oligodendrocytes. The oligodendrocytes provide support to axons of neurons in the central nervous system. They particularly those that have that travel long distances within the brain, right? They produce fatty substance, which is known as myelin, which is wrapped around the axons as a layer of insulation. So, oligodendrocytes are basically the cells that are forming the myelin sheath so that the nerve conduction along the signal conduction along the nerve can occur without loss of signal. So it is acting like an insulation, the myelin that they are secreting, right? So these are the oligodendrocytes. Then there's a third type of macroglial cells, which is known as ependymal cells, right? Ependymal cells line the spinal cord and the ventricles of the brain, and they are involved in creating cerebrospinal fluid or the CSF, right? So there is a region in the brain that is known as choroid plexus that is creating a majority of the cerebrospinal fluid, and the cells that are actually doing it are the ependymal cells, right? Then come the radial cells. Now, what are radial cells? These are basically progenitor cells. That is, they will be the precursors of various kinds of cells, and they can generate neurons, astrocytes, and oligodendrocytes. So, all the major cell types that we have studied just now, they are able to make any one of them, right? So, it will depend on what is the use that will determine their fate, right? So these radial cells are also the macroglia that is present within the brain. Then come the macroglia of the peripheral nervous system, right? There are two such cells, or rather three such cells, if I'm not wrong. One is the Schwann cells. Okay, so Schwann cells is similar to oligodendrocytes in the central nervous system. It basically myelinates neurons in the peripheral nervous system, right? So its function is very similar to that of the oligodendrocytes. Then come the satellite cells. The satellite cells surround neurons in the sensory, sympathetic, and parasympathetic ganglia and help regulate the chemical environment. They may contribute to chronic pain, right? Then the final one is the macroglia of the PNS, is the enteric glial cell. As the name suggests, they are the glial cells that are found in the digestive system or the enteric sac or the stomach, right? So that is that for the kind of for the type of glial cells that are present. Now let us come to the type of neurons, which are the major function or the smallest functioning unit of a nervous system, right? So there are these three kinds of neurons that you generally study. It is unipolar, wherein there will be just one branch that will be coming out of the cell body, right? That is what is unipolar. And this one branch can basically divide into axon and dendrites, right? Then comes the bipolar. In bipolar, what is happening is from the cell body, there are two branches that are originating. One side is the dendrites and one side is the axon, right? In multipolar neurons, there are multiple branches that are originating from a cell body, right? There can be multiple dendrites. Axons, on the other hand, is a single one only, right? Then there are different cell types also that are present dependent on where in the brain they are present, right? So, for example, pyramidal cells present in the hippocampus, Purkinje cells present in the cerebellum, etc. Right? So, uh, again, another kind of classification of neurons can be what their function is, right? So the sensory neurons will be responsible for sensing a stimuli, right? And over here, you can see that generally these uh, uh, neurons are myelinated and they are generally these bipolar neurons, right? Then the motor neurons, which will basically send the response for whatever sensory stimulus has been received, right? So these motor neurons are generally multipolar neurons and they are also myelinated. And then there are interneurons. Now, since interneurons are neurons that are connecting the sensory and motor neurons, and these do not have to travel a long distance, right? So sensory neurons might be bigger neurons, motor neurons might be bigger neurons, but interneurons are only covering a short span, right? Between the sensory and motor neurons, they are generally non-myelinated neurons, right? Then we come to synapse. Now, a disclaimer before I talk about the synapse that is represented in this figure is that this is a very, very simplistic representation of a synapse, right? There are much more components, many more components that are present in the synapse that for the ease of it, we are not discussing right now over here, right? So, this is a simplistic a representation of a synapse between two different neurons. So this is neuron A. This is the axon of that neuron, and this is the dendrite of the neuron B. Right. So from the first neuron come synaptic vesicles, which basically contain these red color substances, which over here are the neurotransmitters. Right. These neurotransmitters are basically what is going to transmit whatever information is there in the chemical form to the other neuron, right? So these neurotransmitters will be released at the ends, the axon ends of the first neuron or neuron A, and they will basically be present in this gap between the two neurons. This gap is known as a synapse, right? This synapse will have a neurotransmitter that can go and bind to the receptor that is present on the dendrites of the neuron B, right? And that is how the signal will be transduced from neuron A to neuron B. So, again, this is a very simplistic representation. There are receptors that are present even on axons of this neuron A that will basically recycle whatever excess neurotransmitter is left in the synaptic region, right? So, once the information has been passed, the neurotransmitters can't remain in the synapse, right? If they remain in the synapse, the information will keep passing, and that is not how it works, right? It is, for example, I just saw something. And I saw the slide that has a synapse and I responded to it, right? Now I can't keep responding to it the same way, right? I have to move on to another slide and I have to respond to a different network, right? This network has to shut down for that. So for that, the neurotransmitters that will be remaining in the synapse will be recycled either by their reuptake on this axonal end by some of the receptors that are present over here for the reuptake, or through enzymes that are present in the synaptic cleft, which will basically degrade these neurotransmitters, right? So that is how the synapse is working. Also, another thing that I wanted to mention over here is that the synapse shown over here is a synapse between two neurons, but a synapse is basically a chemical passage that is delivering, neurotransmitters or chemical information from one from a neuron to the other part across the surface, right? So these synapses are even present between neurons and muscles, for instance, which is known as a neuromuscular junction, or the neurons and skin, right? So there are different kinds of cells that can be involved in these synapses, right? But on one side of it, it has to be the neuron. Yeah. Then this is a representation of the nerve circuit that we also discussed in our assignment, previous year's assignment, and how the brain is not really involved in the reflex action, such as a knee-jerk reflex, because the entire relay of the signal and processing of the signal and the relay back to actually act on the signal is happening through the spinal cord. That is why it is that quick, right? So again, I am not going into detail of this because this is exactly same as to what you have studied in the lecture provided by Sir. So this is just for your recap. Uh, then let us just discuss some of the common terms that are present that are generally used with respect to the brain and the nerves and the certain neurons in the brain, etc. So, what is a white matter and gray matter? This is something we have been hearing from a long while, right? So, white matter is basically made of myelinated axons because of the myelin sheath on top of it. It gives a whitish appearance, and gray matter is made up of cell bodies, right? So, what is a nerve and a tract? A bundle of axons found in the body is known as a nerve. And okay, I think I have messed up over here. So, a bundle of axons found in the body is known as the nerve, but bundle of axons found in the brain is actually known as the tract. Okay. And in case of ganglion and nucleus, group of cells that are found in the body are known as ganglion, while the group of cell bodies that are found in the brain is known as nucleus, right? So that is the difference. I think that, okay, I think this might be one of the last slides that I could arrange for this week's recap. And as I said, out of nine, I think up till seven I had done, and the other two I will include it with the next week's discussion, right? So, when we talk about the nervous system, there is the central nervous system that consists of the brain and the spinal cords, also the cranial nerves that are arising out of the brain. And then we talk about the peripheral nervous system, which basically consists of the nerves that are reaching far across different regions of our body, right? The peripheral nervous system is further divided into the afferent and efferent divisions, right? So the afferent division is the sensory division, whatever is being sensed or whatever is basically getting the stimulus. So sensing the external environment and sensing the internal environment, both of this is performed by the afferent division or the sensory division. And then the efferent division or the motor division is what is actually performing the task on the once this sensory information is uh, achieved, right? This motor division can be divided into autonomic nervous system and the somatic nervous system, right? So autonomic nervous system again is further divided into sympathetic and parasympathetic nervous system. Somatic nervous system is the different organs having different kinds of brain activity, brain control, right? Then come the protective layers of the brain. So on top of the brain are the three meninges that are present from the internal innermost to the outermost. These are the pia mater, arachnoid, and the dura mater. On top of this is the bony layer of the brain, that is the skull, which forms the cranium, and then come the skin. Right. So these six layers actually protect the brain. Okay. Then, because we have been talking about CSF so often, and especially during the historical perspective slide, we talked a lot about CSF and how multiple people thought that this is how the brain is functioning. This is how it is basically taking care of the function that is being performed by the brain to monitor the entire body, right? So what is cerebrospinal fluid actually? Chemically, it is an ultrafiltrate of the plasma that is contained within the ventricles of the brain and the subarachnoid spaces of the cranium and the spine, right? It performs vital functions including providing nourishment, removal of waste, and protection to the brain. So, since it is a liquid that's also surrounding the brain, it is also basically creating a cushion kind of environment around the brain so that from mild impacts it is secured, right? And obviously, there are metabolites, wastes, etc., that are being removed from the brain through their disposal into the CSF, right? Then come the volume. How much of CSF is actually present? Adult CSF volume is estimated to be around 150 ml, with distribution of 125 within the subarachnoid spaces, right? The spaces that are present between the arachnoid and the brain itself, and 25 within the ventricles, the holes that hollow regions that are seen. Right, CSF is predominantly secreted by choroid plexus. What cells in the choroid plexus? Ependymal cells, with other sources playing a more poorly defined role. So we are not very sure as to what other regions might be secreting it, but for choroid plexus, we are definitely sure, right? So that is it for this particular session, and I'll just sign off. Stop sharing my screen and sign off. So yeah. So I hope I share this recording with you guys and with NPTEL, and I hope that next meetings link will be shared with you people so that some of you can also join the session, so that if you are actively actually having any doubts, it can be addressed over here. Um, so that is it from my end. I just stop the recording. Thank you guys.